Hi, everyone. Welcome to microbiology. This is our first chapter, essentially getting into gear. This is a new video. A lot of the, well, maybe all of the videos after this, the main lecture videos anyways, those are going to be old from last semester, and I kind of apologize for that. Not really, because I'm getting a new textbook next semester, and it just didn't make sense for me to re-record all of those. So there's going to be some stuff like clicker quizzes that I kind of go over. Don't worry about it. I mean, I'll go over the quizzes. The information's fine. It's just, we aren't going to be using clickers. I mean, we aren't going to be using clickers. So that's the idea. So just be on the lookout for that. Keep in mind, a lot of the stuff I have set up for this course, you will be opening gradually as you work through it. Some of you guys will look at this and be like, 2 quizzes in the chapter. Man, just think of them as like a 10-question quiz, because they're both 10-question quizzes maximum. So 10-question quiz and a 10-question quiz split up by like a review video. So just look at it as a 20-question quiz that has a study opportunity in the middle of it. Just think of it that way. So just be sure you get all the stuff done by the deadlines. I don't have strict deadlines on like each and every single assignment. Everything's due like before exams, but you have to be on it, man. If you let it go for too long, you're going to have way too much to catch up on. But yeah, so, and I did set it up for the quizzes to be easier on these earlier chapters so that you can get into the groove of doing everything. I didn't want to throw you into the deep end with some really difficult questions right off the bat. So as you get through closer to the midterm, you'll start seeing more difficult questions. That'll be a little bit more similar to what to expect on the exam. So that's that. Most of the stuff will be gone over in the syllabus and everything like that. It's all covered. But I just know that most of you guys would probably read the syllabus. So I don't know what to tell you. Yeah, so the midterms and the midterm for the lecture portion and the final for the lecture portion. Those are the only 2 exams that we have for the lecture portion. Because it's an 8-week course, it's shortened for the summer. I didn't want to have like 1,000 exams that were worth a lot of your grade. So I just have 2 exams that are worth a lot of your grade. It is what it is, and that's how it's been split up. Actually kind of works out to your advantage the way that I have it set up. So of course, if you have any questions through any of this, feel free to email me. You can call me. I'm not as good about getting back to people if they call me. So please, I prefer email. You can email me through Canvas or with my actual email address that's available on the Canvas site under the instructor area or in the syllabus as well. So be sure you actually read the syllabus. It's kind of got some important info. And if you have any questions, let me know. If you have any questions, let me know. If you have any questions, let me know. Don't just sit there confused about life and not letting me know. So, moving on. Let's go over chapter 1. That's what we're here for, right? Why is this not going away? Maybe you can go away there. Yeah, I know you guys probably can see it's the little drop down menu just sticking down. So this is chapter 1, which is just introduction to microbiology, kind of what it is and terms associated with it, so that we can kind of jump in a little bit later. So we're orienting ourselves with the so that was my brief orientation. That's all you're going to get. There's other stuff to help get you guys a little bit more situated. And of course, talking in person in the lab whenever we first meet, which for you guys, our first meeting is going to be on the Wednesday, the 7th, and there'll be an announcement about it and everything. So it'll remind you guys, but yeah, that's when we meet in lab. So that's, I'll see you then, I guess. Right. So we're going to talk about what microbiology is. We're talking about cell theory and germ theory, very important theories as far as understanding kind of how diseases work and how, the things that cause diseases work. Some major contributors to the current understanding of microbiology, old guy stuff, and then scientific names and taxonomy and stuff. So microbiology, organisms that are too small to be seen with the naked eye and have to be magnified sometimes. Usually we might define it as organisms that have to be viewed through a microscope So we also refer to them as germs, viruses, agents, bugs, sometimes not really using the terms totally accurately, but I'm a firm believer that for the most part in the English language, if I know what you're talking about, then you're speaking correctly. All right, microbiology is the study of bacteria, fungi, worms. The immune response is included with all this, as you have to understand it to understand disease and our response to it. Are you guys all right back there? Sorry, my dogs are just like having a field day back there. And then viruses. And so most of these are pretty straightforward. I would be aware of that term for fungi is mycology, myco. If you guys played any Resident Evil games, and maybe you know about mycelium, so maybe that won't be quite so confusing for you guys. I'm not very good with things. Sorry, I don't get out a lot. And then helminthology, that is worms are helminths, and we'll talk about them a lot more later on. What is going on, dude? Okay, microbiologists will study cell structure and function. So talking about cell structure and function of like mostly bacteria. Oh, let's be honest, that's most what we're going to study here, but we will be including eukaryotes as well and talk about what those are. Don't worry, that's chapter 5. So cell structure and function, growth and physiology of organisms, microorganisms, the genetics of them, taxonomy, evolutionary history, interactions with living and non-living environment. I mean, anything you can want to understand. Most of this, some of this stuff is really boring to me, like environmental stuff. I mean, I'm in here for the medical side of it too. I think that's what got me interested in, and I know that's probably what you care about too. So I'll try to make this course as interesting as I can for you and not just like learning and the books and the words and all of this, right? So there's a lot of different types of microbiology that we can break it down further into how people want to understand what effect these microbes have on different aspects of our lives. Environmental, like I said, boring to me. Agricultural, it's actually pretty important because we're having issues with the climate now as far as nitrogen in the soil, and plants aren't happy with that. What provides the plants with the nitrogen in the soil? It's bacteria in the soil. And so they're being affected by the climate. And that's actually a big deal. That can actually lead to some countries starving soon. So that's a big issue. If you haven't heard about it, you should probably look into that. Follow the news a little bit there. So, and then industrial is utilizing, bacteria and other microbes for the production of things, whether that would be, medical. Are you okay? Whether it might be like things like insulin or whatever, and that sort of thing. So in medical microbiology, that's not necessarily talking about like making insulin, right? Medical microbiology, that's the good stuff. That's the diseases. That's the stuff we're here for. Don't worry, we'll get into it soon. But you have to understand a little bit about them in order to get into anything. So just saying. The organizations that microbiologists might be relating their work to. It's a weird way to put it, but anyways, the CDC is an obvious one. Centers for Disease Control and Prevention, World Health Organization, and the USDA. All of these institutes have some sort of role and some sort of aspect of bacteria and how they relate to human society, right? Even including in our food, like we have to do with the FDA. And that could be as far as preventing, looking after food safety, making sure you don't have any contaminants in there that shouldn't be there that make people sick, or even just making sure that the microbes that you're using for food, like for making beer or cheese or bread or something, making sure that, everything's in order with that as well. Cell theory. So this is a big one. So how do we categorize something as being alive or not alive? So we're going to go by the concept of cell theory for this course. Basically, all living things are made of one or more cells. If it's not made up of one or more cells, we will consider it not alive. All cells come from pre-existing cells in order to be considered alive. All cells contain DNA in order to be considered alive, and all cells contain a membrane in order to be considered alive. Viruses do not fall under these categories. Some viruses don't have a membrane. Some viruses don't have DNA, they have RNA. They might have single-stranded instead of double-stranded DNA. They make it weird. They do weird stuff. They are not cells, and they did not, well, they came from cells, but they're not cells that came from cells. They are viruses that stole parts of the cells and hijacked the whole cell system in order to make their parts and put them together. So viruses, we are not considering them alive. So definitely remember that. Viruses, they're not alive. They're not dead. Dead is for if you were living, right? And they were not that. They're just not alive. Never were. All right, microbes, we know that they're everywhere in the soil. They're probably on my Chick-fil-A cup right here. I mean, they're all over the place. So definitely on my keyboard, definitely on my dogs. They're gross. I mean, they're dogs. They're not like particularly gross, but so they're also all over in the news, right? We've heard all about COVID. You've probably had COVID. I had COVID. That's everywhere. Or we've talked about in the past Ebola or flu when we have outbreaks of these things as well. So we watch them in the media. We know about in the environment, there's a little bit of a talk of whiskey mold as far as Jack Daniels. Recently, I was talking with Dr. Rhodes about this. Jack Daniels, essentially, and their plant in Tennessee, they were, letting their whiskey sit in their barrels. And there's what they call the angel's share, right? It's the part of the alcohol that evaporates out of the barrels. And anyways, there's a mold that likes this situation and it grows in that area. They call it whiskey mold, and it has like covered the whole town of where this is. And I'm not kidding. Like people are having to power spray their houses off like every couple of weeks because there's so much of this mold. And it's like a black colored mold. It's not black mold like the scary stuff, but it is a black colored mold. So it's pretty interesting. And of course, there's going to be a lawsuit involved in that. But anyways, and then when we're talking about evolution, which evolution couldn't be more evident than it is in microbes like viruses and bacteria, in the development of drug resistance and discovery of new strains or development of new strains due to, pressures from growing up in certain environments. Talking about the microbes, of course. It's really interesting stuff, and it's just exciting how much there is yet to learn about these guys. So a disease is any situation where you're feeling from normal, a deviation from normal. So if you're not feeling well, we would consider that disease. It can be microbial or non-microbial, right? Bacteria, strep throat or something like that. Microbial, you could have, breast cancer. That's not microbial. So that's a disease as well. So just remember, it's not all about microbes as far as disease goes. There's other kinds. In this course, of course, we're going to be talking about microbial related diseases. We can say too, we're talking about like disease and what it is and how like communicable diseases work. Some diseases are caused by microbes, right? Some that aren't, cardiovascular and cancers, things like that. We just talk about that. But the idea is if it is a communicable disease, like I get sick and then I pass it on to you, and it, shown essentially, and we'll talk about how in a little bit, but it can be shown that it is passed from person to person, then that is what the germ theory is as far as the cause of disease. Germ theory refers to communicable diseases have to be caused by something, microbes, pathogens is what we would call them, disease-causing microbes. And we'll get into that soon. So a pathogen, that term that I keep using, that is any agent, any microbe typically, that includes viruses, that can cause disease. So when we talk about microbial causes of disease, we are talking about pathogens. There's over 2,000 that can cause disease. So how are we going to prevent disease? Primarily we use vaccination, right? And the whole point of vaccination, training our immune system so that when it sees the thing again, it's sort of like you trained these specialists to recognize this particular bad guy. And when that particular bad guy comes around, they're already ready to go. Took a little bit to train them up, but now that they're trained, they're ready to go every time that they see them again. That's what vaccines are about. I'm not going to get too much into the anti-vax movement, but I will say this. This course is pro-vaccine. If you want to have a discussion about your anti-vax mindset, I'm happy to do that, but I don't want you necessarily discussing it in class because I don't want it to be incorporating people's thoughts about, the course and what we're trying to teach in the course. What we're teaching in the course is pro-vaccine. So if we look at these figures here, I want to point out polio in the US, and I want to point out smallpox. Smallpox is caused by a virus called variola virus. It's a horrible, disfiguring disease. It really is. And there was a man, Edward Jenner, that discovered that cowpox, whenever you're exposed to that, it actually prevents you from getting sick to smallpox. And it's a different vaccine. That was vaccinia virus. And that's where the word vaccine comes from. That's essentially the very, very first vaccine ever. And all of the rest of them were based off of that concept. It's pretty cool stuff until you learn that the guy did the experiment on his kid. So I don't know about that. Anyways, vaccination against smallpox has eradicated it from the planet, which is pretty awesome, guys, considering how much of a problem it was. Yeah, no, we don't have it anymore. It's only kept in cold storage in certain countries, the United States and Russia. So, but yeah, I think that's pretty cool. We're on the verge of eliminating polio. The only places that we are seeing polio pop up in the world now is Afghanistan and Pakistan, just having some issues getting full vaccine coverage in those areas. But that's the only places in the world where you see it. It's almost completely eradicated from the world. And I think that's great too. Another horrible disease. It exists only in humans, and obviously it puts people in wheelchairs, paralyzes them, and all this stuff. It's a horrible, horrible disease with lasting awful effects. So I think it's great that we're moving in that direction. We have vaccines to thank for all of these things, and how can you not love it? It's a great time to be alive, except for COVID. I mean, we have vaccines for it, but all right. The recurrence of measles, because people are choosing not to get vaccinated because of their anti-vax beliefs. We have massive outbreaks of measles now all over the United States. This is something that if 95% of the population in the United States, well, anywhere in the world, if 95% of the population were vaccinated against the measles, the other 5% would be completely protected and we could eradicate it. That's called herd immunity. But measles is so communicable that you have to have 95%. You can't have 50. You can't just be choosing not to, because then we'll have maintaining that illness in the population. That's how you eradicate something. You have to actually get rid of it. You have to be vaccinated against it. So, and measles is one of those things that you can get it and be sick with it and not actually keep immunity against it. It's just one of those sicknesses, that's weird saying, those, diseases, it's a virus that you don't always, when you're naturally exposed to it, get immunity against it. Actually, really not uncommon for that to happen. So vaccines can be really great. We'll talk more about vaccines and the immunology, because I could go on forever about it, but we will talk more about them later when we get to that stuff. So humans have been using microorganisms for thousands of years. Bread, wine, cheese, all of these things have been used to make really great, tasty foods. They've been doing it since, I guess, Egypt has been using moldy bread to treat wounds. They basically discovered penicillin, but they just didn't even realize it. That's awesome too. So that's a lot of times because mold will produce things like penicillin to keep bacteria from coming in and encroaching on their territory. So it's defensive, really kind of cool stuff. So we're going to talk about prokaryotes and eukaryotes soon. And I know these are big terms and big words. A prokaryote, pro being before. Karyote, think of karyotyping. That's like whenever we take the chromosomes from people and try to determine like how many they have and count them and line them up and all that. That's from the nucleus, right? So prokaryotes before nucleus. They don't have a nucleus. They don't have a true nucleus. Their stuff is just hanging out inside of the cell. There's no nucleus. And then we have the endosymbiotic theory. The idea there is that we had a cell of some kind and then a bacteria that was really good at making its little energy. And this guy ate it up and now it's in here And then that was just a happy situation for both of them. The inside guy, maybe it got protection and got other food, and it was just making a molecule that this cell could use for energy. That's pretty cool. That's the idea of where we got our mitochondria, which we'll talk about that in chapter 5, because we are eukaryotes. A eukaryote, eu means true. So we do have a true nucleus that holds our genome in a membrane. So prokaryotes, they don't have any membrane-bound organelles. Organelles are like kind of like what they sound like little organs inside of these cells. Yeah, eukaryotes do. So we get all these fun things like mitochondria and the endoplasmic reticulum and everything. Chapter 5. All right, this is a picture of stromatolites. These are super, super old stacks of just biofilms of cyanobacteria. Cyanobacteria just use sunlight for energy. They're like kind of like plants, but they're bacteria. And these guys can be like, around for billions and billions and billions of years. It's crazy. So yeah, we think they've been around for a very, very long time. So cool. All right, the scientific method. This is where we're going to be, forming our hypothesis about some sort of problem. We think, I think that these people are getting sick because of a bacteria, and like how we're going to figure it out. So that would be a formulation of a hypothesis, some explanation for the problem. And then we will use something called deductive reasoning to apply. We will apply deductive reasoning to determine, whether we're right with our hypothesis or not. So that means we are going to form our hypothesis. We're going to conduct an experiment. And if that result is aligned with our thoughts of our hypothesis, then we will accept the hypothesis. A lot of times in research, why I hate research and why I don't do it now is because most of the time it's not going to line up, because most of the time you're not a genius and you didn't get it on the first try. So you have to keep going back and changing a little thing and changing a little thing. Oh my gosh, it is boring and depressing. So it's not for me. Any of you who want to go out into research, kudos. So yeah, you're basically testing a theory, come up with an idea to explain something. And I do want you guys just to be aware of that idea of the deductive approach, that you make an observation, you ask a question, you form a hypothesis about what you think is going on, and then you test it to verify it. With inductive reasoning, that is where your observation leads to the theory. That's like what Sherlock Holmes does. So he just kind of is just observing and making theories as he goes along instead of going and testing them, right? Right, in nature, what do microbes do? We already know. They are involved in making your food rot. They're making everything else rot. They're breaking all the dead things down. 70% of photosynthesis, this is a good one, is performed by microbes. And that might be crazy to you. You're like, what, the plants? I'm sorry. I just showed you the bacteria in this. So in this other picture, these cyanobacteria, again, these are basically photosynthetic. They're not basically, they literally are photosynthetic bacteria. So they're making oxygen for us. All of this stuff out here, this green nasty out here, that's algae. And what do you think they do? Photosynthesis. And what are they? Are they plants? Well, I'm going to tell you right now, they're not. Maybe you don't know. They're not plants. Algae are belong to Protista kingdom. They are not plants. So microbes, they're considered microbes. Pretty cool stuff. That's not plants. Yeah. And then a lot of microbes have developed symbiotic relationships with plants and animals and with each other. That just means that their lives are intertwined somehow, and they've found ways to work together, depend on one another. Sometimes like kind of permanently, like a permanent kind of relationship that their lives may depend on, sometimes just in passing, right? So, and when you talk about, sort of talk about, breaking down the dead things, decomposition. So that's some bacteria and a lot of fungi are involved in that, right? So that's how we are going to recycle the nutrients back into the, environment. So germ theory is that idea that communicable diseases are caused by pathogens, microbes that cause disease. Louis Pasteur kind of came up with this idea. People were saying of spontaneous generation, that if you lay a meat out and like you can see my hands, if you lay a meat out and then, you know, a day or two later, now there's maggots on it, but it's spontaneously generated. Like it just came out because that's what happens when meat sits out. Not, it's not. What it is a fly came, right, and laid some eggs on there. Or like your milk is spoiling, not from sitting out, not because of spontaneous generation. It's because of there was already bacteria in there, and now they're multiplying because you weren't keeping it cold anymore, and now they can grow at room temperature. So that's the idea there. And that's why I bring up, because Louis Pasteur, who came up with the concept to get rid of spontaneous generation, things have to come from other things, right? That's the idea with the cell theory. That's just bringing it over to disease. He's the one who came up with pasteurization. It's named after him, which is just heat and cold over and over again to kill off pathogens, like in milk. So yeah, that's the germ theory of disease. Robert Koch was this guy who came up with these postulates to verify that. So basically, he said that we have a disease-causing organism, what we think is the disease-causing organism. Let's say we, separate it out. So we have a healthy animal, wherever we're collecting this sample from, they don't have it. So we look in the microscope or we look on the plate or whatever it is, and we see these, black rods here. And so we think that maybe that's the pathogen that this animal or person had. And so we can isolate it, basically. And this is a whole series of steps. Culture it, meaning we're going to grow up the bacteria so that we have enough to work with to do our, studies. But basically, we're going to isolate it in his theory and get it out to where we know we have just that pathogen and not a mixture of things. If I swab your throat, you have more than one bacteria in there. I'm just saying. So we can isolate it and then inoculate or put that organism into a healthy organism. And if it gets sick with the same thing, and then we can re-isolate it back out again, it's the same thing again, that's confirming that that is the causative agent of that disease. So these are steps that we would follow to try to discover causative agents or microorganisms that are causing disease, pathogens for that disease. Cool stuff. Okay, microbial classification, taxonomy. This is, I know you guys feel the same way, sorry, but I do have to talk about it. So taxonomy is classifying living things. It's Carl von Linné who started doing classification of these things, or Linnaeus is another way that some people say it, just depends. I don't really, as long as you can see it on a paper and choose it, that's really the idea, right? So, or online. Right, so whenever we are naming something, we name something via the, binomial nomenclature. So that is binomial, bi being two, noma being name. So it's got two names. So the first name is always the genus and it is capitalized. The second name is the specific epithet and it is lowercase. That's always how it is. So like Homo sapiens, you capitalize the H and lowercase the S. That's how we would write this out. So you guys have seen this stuff before. Always italicize it. So if you guys are typing up a paper or filling out a form online for me, please be sure that when you write, species names like E. coli and stuff like that, that you are italicizing and capitalizing the right things. If you need to refer back to this or ask a question about it, it's fine. It's part of what can cost you points later on. I'm just warning you. So italics and capitalization are important here. So what constitutes a species and how do we define that? It's like impossible. I don't even know how to describe it. More or less, they're mostly basing it on genetics, but they are also considering other characteristics of organisms. And that's how they're going to determine what categories to put them in or whether or not they're related or whatever. This is all just really, guys, this is all just man-made BS. So I'm just saying. Okay, naming examples. Here we have Staphylococcus aureus. Staphylo means bunch of grapes. Coccus means berry. So that's the round sphere shape. Aureus means golden. That's what it looks like whenever you have it out on like a plate, and you get those little colonies. We call them colonies, little bumps that you get on the plates when they grow. So that's just descriptors of what it looks like. Campylobacter jejuni. Campylose means curved. That's what the cells look like. Bacterion, bacter, little rod. Jejunum is part of the intestine. That, this is a organism that causes diarrhea from eating spoiled food or something. I would say spoiled food. That's really accurate. It's not spoiled. It's just, people not washing their hands right. And then we have Giardia lamblia. This is a protozoan. This Giardia part is named from Alfred Giard, who's a microbiologist, French, I guess. And then lamblia comes from Victor Lambel, who is a Bohemian physician who were involved in identifying the agent that caused the diarrhea from the water. It's like if you drink creek water or something. Yeah, I'm sure you know what happens. You can imagine anyways. So, they get a little bit creative with their naming, with chaos, or ahaha, or abracadabra. Some of them get pretty creative and they're not based on scientific aspects of the organism. And that's fine. That's probably how I would name an organism. Probably name it something ridiculous like this. I don't know. Anyway. Anyway, it's silly. Okay, so the first step to classifying things, we're going to get into talking about domains here in a second. And then first you have to understand about eukaryotes and prokaryotes. We mentioned it a second ago, but repetition saves lives. So I'm really big on that. So you'll hear me repeating a lot of stuff over and over again. That's for your benefit. Eu being true, kary being nucleus, organisms with a true nucleus and with membrane-bound structures in them. That is eukaryotes. And we'll talk about it. Don't worry, we'll talk about what a membrane is next chapter, chapter 2. The chemistry chapter. You didn't have enough. Prokaryote, that's pre-nucleus. That's bacteria and archaea. Okay, so bacteria and archaea are the prokaryotes, both of them. And then we are eukaryotes, so, animals and plants and fungi and the protists. All of us are eukaryotes, excuse me. So, so bacteria and archaea, prokaryotes. The ones that we would consider, typically have come before us in evolution, which just makes sense, right? They're unicellular organisms and they're a lot simpler. So prokaryotes, archaea, and bacteria. Archaea are basically like, they look a lot like bacteria and kind of behave like bacteria, except they live in the extreme environments. And their genes and stuff seem to resemble, and they have traits that are a lot more like eukaryotes than prokaryotes. They kind of don't no, you can't really classify them the same way. So that's why we have the domains split up the way that they are. So our domains would be split up into prokaryotes. What is that? Beeping. Prokaryotes. Oh, it's my email thing. Eukaryotes. No, that wasn't right. Sorry, that beeping threw me off. I'm sorry. That was. Okay, so the domain. So we know we have the prokaryotes and the eukaryotes. Eukaryotes, fine, they're on their own. The prokaryotes. Now, I told you that bacteria and archaea kind of look the same, but really the properties are not very similar. They have different stuff going on in their membranes and walls, which is what is going to hold everything inside of the cell. Very different there, very different genes. They live in these really crazy extreme environments, like down in the deep sea vents and stuff. So those are our archaea. So our 3 domains. I'm not going to write this all out because this is ridiculous with my mouse. I forgot to bring my writing thing, tablet, home. So I'm not going back to the university. It is, between semesters. I'm not sorry. No. So bacteria, archaea, and eukaryotes. Those are the domains, just those 3. That's all that you need to know of them. So we're going to classify them based on similar characteristics, like literally their behavior and growth requirements and stuff like that, as well as mostly genetics. And that way that we are, organizing these things based on their genetics, that term for that is phylogenetics. Yeah, so that's that. So that's how we're going to sort them out genetically, and then we'll also consider some of their characteristics. Any common ancestors and things like that. So these are the levels of classification. You'll probably be quizzed on this or tested on your exam. So I remember these: kingdom, phylum, class, order, family, genus, species. Don't worry about division. That used to be what it was called. We're going to be calling it phylum. And you don't really need to worry about these as much, but that is how we would subclassify if we needed to. But yeah, kingdom, phylum, class, order, family, genus, species. You've probably heard this before, maybe in high school. Make a little acronym for yourself so that you can keep them in order, because I will ask you to put some in order or recognize which ones are in the right order. For phylogeny and trying to separate things out based on the DNA, we use the 16S ribosomal RNA. Basically, that's part of the ribosome, which is a little thing inside of the cell that helps cells make proteins. Proteins pretty much do all the work in the cell, I'm just saying. So it's like enzymes and things like that. So ribosomes are like the tool the cell uses to make them, proteins. And all cells have to have ribosomes, all of them do. So we will sequence based on their similarities in their ribosomes, since we know everything has that. And then use similarities and differences based on that. And that is the basic idea behind phylogeny and how it works nowadays. And that's how we have separated out archaea, bacteria, and eukarya. Those are our 3 domains. Carl Woese was the one that came up with this concept to use that the genes for the ribosome, essentially, which is pretty creative since everything has to have one. Right. Oh, that's it. So good. So that's chapter 1. Be sure you read the syllabus and take the quiz. There is a limited time on that. You can't just leave it till the end of the semester. I think it's like 2 weeks. But I mean, Canvas will notify you on most of this stuff. Just be sure you work through each chapter systematically. It's set up that way on purpose. So try to follow through with it the way it is set up. There are optional things on there, of course, things like study guides. Like, I'm not giving you credit for those things. You don't even have to do them. You don't have to do the Kahoot quizzes if you don't want to, and you don't have to participate in reviews or any of it. You do what you need to do to learn best from the class. I'm not going to tell you what to do outside of, I'm not going to tell you what to do. That's just the bottom line, right? I might have assignments that have grades that you'll get zeros on if you don't do them, but that's me doing me. So I get to do what I want to. Turns out we're both people, and that makes us equal. So read the syllabus, complete the quiz before I get too off track again. Remember, microbiology is multidisciplinary. It's a study of microorganisms, too small to see with the naked eye. They're everywhere. Microbes, scientific names, 3 domains, microbes, right? So that is it for chapter 1. I believe that's everything. Yes, it is. Wow, that really, like, look at me now. Where did I go? The screen was lighting up my face. So I'll go back to that. Yeah, let me know if you have any questions. I'm going to go ahead and call this a day and get this uploaded. And then, hey, I'll see you in the lab when we get there. Bye.